students, welcome to Vedantu NEET English. I am Ms. Gopika, your biology master teacher. I hope I am audible, visible and the presentation right behind me is also visible. Please let me know in the chats if we are audible, visible and you can see me live. Yes. Hello, good morning students. How are you all? We are in good afternoon, right? Okay, good afternoon students. How are you all doing? Are you all doing great? Because it's been some time that we met. Hello. So those who are new, quickly smash the like button and mark your attendance, and also subscribe to the channel because this channel gives you the best content possible in English. Hello, yes. So I think we are live. Awesome. So students, we are going to do locomotion and movement. I think one chapter that students make it very tricky in their head, but it's actually a very easy chapter. So today I'm going to decode that chapter. So today we are going to understand locomotion and movement part 1, where I will be discussing actin, myosin, sliding filament theory, very important topics of this chapter. And sometimes we can also have more questions from this chapter. Okay, so every year the questions vary from, let's say, 3 questions to 4 questions. Okay, 3 questions to 4 questions is how much it varies. So that means you can expect somewhere between 12 to 16 marks. Okay, 12 to 16 marks from this chapter. And let me tell you, this chapter questions are usually on the easy end. Okay, they are not difficult, they are on the easy end. This way you will be able to easily score that 12 marks. So let's do it. Okay, let's do it. Awesome. So those who are new, I would request all of you to smash the like button. Now students, locomotion and movement. Let's go to the introduction of this chapter. All of you stand up. Wherever you are standing, just stand up and just do this. Okay, just do aerobics. We are going to do a Zumba class. Okay, yes. If you do like this, I will say it's a movement. But why, on what basis did I tell it is a movement? On what basis is that you did not have any displacement. You stood in that position and you moved your arms. Right, you moved your arms, you raised your hands, but you did not locomote. That means you did not move from one position to another position. But just imagine we moved like this. Okay, I moved and I came back again. So this is locomotion. That means, very simple students, moving away from the original position to an organism is called as locomotion. That means maybe we are moving to take food, maybe we are moving to drink water, maybe we are moving to do something, but we are moving from one position to another position. That would be called as a locomotion, and it is completely voluntary, right? If I decide, okay, if I decide to go take my NCERT book, then only I'm going to study. So it is my decision. Now let's say movement. Movement can happen with or without moving away, with or without moving away. I can stand here and maybe try to grab something, or I can stand here and do some movement. Do you understand the difference? Okay, so that means locomotion means where you are doing some kind of a displacement. Some kind of a displacement is happening here, but at this place displacement is not necessary. Okay, displacement is not necessary. Is not necessary. So I want all of your focus here because we are going to use some videos to understand. I have kept everything ready for you, so everything is like, easy peasy in your hand. Okay, yes, let me also check your attendance in that time. In meantime, let me check your attendance. All of you mark your attendance because that is one thing that you can do to ma'am, right? Yes, very good. I want the energy that Hasini has, Madhumati has and Divya has. Okay, that's the kind of energy that I'm expecting from all of you in the class, because after this class this topic will be just flowing, right? Everything will be so easy. Okay, so now you understood the basic difference between locomotion and movement, but there's a very important statement from NCERT which is also a very confusing statement. Look here. All locomotions are movement, but all movements are not locomotion. Yes, ma'am, absolutely right. When you did the first movement, you did not move, but you call that a movement. So that means all movements need not be locomotion. Okay, all movements are not locomotions, but all locomotions are movement, because if I have to that means if I have to do something, I will move, I will do some kind of a movement, right? So this is one statement. Okay, don't have to take any tension. They will hardly ask you maybe assertion, reason or something like a statement based question, but that is also a very rare chance. Very good. Yes, now the energy is lit up in the chat box, and also mark your attendance and only leave the class. Okay, now students, we always tell that movement, the first thing that comes to our head is pseudopodia, okay, cilia. right? Flagella, these are the things that come. So in our body also there are different kinds of movements. First one is amoeboid movement. Now look here, this is how your amoeba will be, okay, a beautiful amoeba. Now this amoeba has pseudopodia, that means false feet. Yes ma'am, they have false feet. We have been studying it from the small class. The false feet is called as pseudopodia. Okay, it's called as pseudopodia. Now, why are we learning it in locomotion and movement under human physiology? The answer is that in our body also some of them does amoeboid movement, students. That means some of the cells are like, okay, wait a minute, I can move like amoeba, I can act like, or I will think that I have pseudopodia and move like that. Same way, in our body also some of the cells does amoeboid movement. One of the biggest examples is that if you see, these are your capillaries, okay, these are your capillaries. So in between these capillaries there are small pores, okay, these small pores are there. And your WBC, which is flowing in your capillaries, will ooze out, okay, which will go like this, okay, let's say this is your WBC. They will go out, move out of the pores and go, and let's say one virus or something, one foreign particle has affected this tissue, okay, this is the affected tissue, students. This tissue, to save this tissue, to protect our body, what will WBC do? It will move out, or it will squeeze out of the capillary pores, go and do phagocytosis of this foreign particle. This movement is an amoeboid movement, and this whole thing is called as diapedesis. Very good, Hasini, diapedesis. Okay, that means where the WBC squeeze out of the capillary pores and go and attack the cells and do phagocytosis is called as your amoeboid movement, is shown by your WBCs and your macrophages. Okay, very, very important. This is called as diapedesis. I had taught you this in your body fluids and circulation, so you should be already knowing this. Okay, diapedesis. So amoeboid movement is shown by two main cells. Okay, I'm writing here example. Okay, two main cells, that is your WBC and macrophages. Okay, macrophages. So both of them will be able to do phagocytosis where they engulf. Okay, where they engulf something. So same way, now let's say they're engulfing this, okay, they're engulfing this, they'll engulf like this and they will swallow it, right? So this engulfing is called as phagocytosis, phagocytosis, okay. So this is a complete amoeboid movement. I hope it is clear. Yes, all of you, awesome. So diapedesis, yes ma'am, understood. WBCs and macrophages, WBCs do diapedesis, but WBCs and macrophages do amoeboid movement. That means they move just like your amoeba and they do phagocytosis. All of you would have seen phagocytosis, and WBC also is irregular in shape. They're not like biconcave, none of that shape, no. So they have a similarity to do amoeboid movement. Now next one is ciliary movement. We have all heard ciliary movement, ma'am, cilia, hair-like substance, hair-like thing, right? Yeah, Madhumati, we can't do anything. For me also ads come. Okay, for my own class ads come. Now think what we can do. We have to take premium. YouTube is telling us use some money, spend some money and buy premium. Okay, that's what we have to do. Yes. Some more people have missed to mark your attendance. I know them. Okay, if I catch hold of you, I will call you conjuice, so mark your attendance. Now students, next one is ciliary movement. We have all heard the word cilia, right? Hair-like substance, okay? Hair-like substance. Now this hair-like substance, what would they do? Very simple thing. Imagine these are your tubes in your body, okay? These are the tubes in your body. In these tubes, let's say some hair-like structures are there. Okay, I'm drawing some small cells, okay, and they have like this hair-like substance. Now this hair-like substance will do a very similar job, that is to move something, right? Movement of something. Let's say ova is coming, okay, let's say ova is coming here. To move this, it might help. To push something, it might help. So ciliary movement is mainly seen in your tubules, that is inside any, say, respiratory tract, fallopian tubes, lining of your fallopian tubes. In these places you will see ciliary movement, that means hair-like substance. Now we have studied columnar brush border epithelium, cuboidal brush border epithelium. What are they doing? They're pushing something, right? They're moving something. So that same movement that cilia shows will be shown in our body as well. Okay, that same movement will be seen in our body, especially in the linings. Linings of what, students? linings of fallopian tube, okay, fallopian tube, linings of your respiratory tract, okay, lining of respiratory tract, all these places you will be able to see ciliary movement. That means something is moving, some movement is happening, and because of hair-like substances are there, this is able to move. Okay, so this is the example of that. Now next one is muscular movement. Ma'am, muscular movement. Now what is muscular movement? Very simple. Movement of your jaws, right? Movement of your limbs. All this is muscular movement. Your muscles have to contract. Yes, respiratory tract, your fallopian tube. Okay, I've written no. Yes, respiratory tract. Even some in your vasa efferentia to do the movement. So all this is there. Okay. Now students, very important thing, muscular movement. Muscular movement is where if I have to contract, my muscles have to contract, my contractile protein have to decide to contract. So all that is there. No, so same way muscular movement. We have around how many muscles? Can anyone tell me in the chats how many muscles do we have? Now muscular movement can be seen in your jaws, okay, limbs. These are the places where you see muscular movement, right? Where you see muscular movement. Tell me how many muscles do we have in our body, students? All of you in the chats. Okay, all of you in the chats. Muscular movement, where do you see, or how many muscles do we have? Yes, you can all tell. Very good, very good. Okay, very, very good. We have around 639 muscles. Okay, 639 muscles, and these muscles are the main reason for us to do any kind of contraction, any kind of relaxation, everything, say tongue, say jaw, say to your limbs, all of these movements are called as muscular movements. So all you have to remember is ameboid movement, that is done by your WBCs and macrophages, ciliary movement, where you can see in the lining of our body, lining of your fallopian tube, lining of your respiratory tract. This is what you have to remember. Now one thing which is not there in your textbook but would be asked in your NCERT, I mean in your NEET, is your flagellated movement. Okay, what is it? Flagellated movement. How can we forget flagella, students? Whenever we talk about movement, we always tell flagella. Now flagellated movement, very simple. Now you can see it in your euglena, right? Flagella is there, it is pushing something, it is moving somewhere. So it can act like a tail, it can act like something that will push everything, move it apart. Same way in our body, that is especially in your, no, only in your males, right? Sperm has a flagellated movement, okay? Sperm is the only one that shows flagellated movement, okay? Sperm is the only one that shows flagellated movement. That means the tail will help them, right? The tail will help them to move and push, right? Let's say your, okay, yeah. Tail will help them to push everywhere, and because of that flagellated movement is shown by your sperm, which you can very easily remember. It's not that difficult, right? So we have done ciliary movement, muscular movement, flagellated movement, and all these notes will be available on Vedantu NEET English Telegram. And if you want the links of all my class and you want to talk to me, then you have to join my Telegram, that is Biology by Gopika. Okay, chalo. Now students, very simple things is over, introduction is over. Okay ma'am, introduction is over. Now we are going to get into the actual part of the chapter, that is properties of muscles. This whole chapter is about what? Muscles. Now today we are going to learn what are the properties of the muscles, what are the contractile proteins that is helping in contraction, and in the next class we are going to study about the skeletal system. Chalo, let's start. Now students, properties of the muscle. First of all, we should tell muscles are mesodermal in origin. Okay, mesodermal in origin. And our body constitutes around 40 to 50% muscles. Okay, 40 to 50% muscles. Now, properties of muscles means what? How did these muscles are able to contract, relax, they're able to stretch? What are those properties? Now, let's understand the properties of muscles. So, properties are only 4 of them. Okay, very easily we'll understand. Don't even worry. Okay. First property I'm going to tell is excitability. excitability. Now what is excitability, ma'am? Let's take an example, okay? Evolution part 2 notes I've shared. No, Hasini? Have I not shared? Don't worry, after this class I'll share it, okay? In case I've not shared, I thought I shared it. I'll check it once again, okay? Now students, excitability. That means a muscle has to take the impulse and do the job. Let's take an example, okay? We have Grace in class, we have Hasini in class. Both of them have ordered coffee, okay? Both of them, let's say Grace has kept the coffee, Hasini went and touched the coffee. Coffee is hot. Immediately the signal that has come from the brain is to retrieve your hand or remove your hand. Now imagine the muscle does not get excited by the nerve impulse. Then obviously the muscle is not going to contract. So this is the muscle, right? This is the muscle. Striations are there, okay? Striations are there. This muscle has to get excited from the nerve impulse, okay? This is your axon terminal. This excitability, the ability for them to take the impulse and do the action accordingly is called as your excitability, okay? Got it all of you? First thing is excitability. Okay ma'am, understood. Next one. What is next one? Second one is elasticity. I'm not going in your NCERT order, but elasticity, anyway, all the four you have to understand. Elasticity means what? The ability of the muscle to stretch a little, right? That flexibility, okay? Elasticity means that flexibility for the muscle to stretch a little. I'm not telling from here I'll go and touch the monitor there, not like that, but the ability for the muscle to stretch a little, that's possible, right? Now if you have seen, let's say India's Got Talent, if you see some people know who is getting the dance and all that, those people have the ability to, they're very flexible, right? If you would have seen the dance, very flexible they are. That flexibility comes from a lot of practice. So muscles are actually that they can retain that flexibility and do a little of like elasticity is there, okay? That is your elasticity. Just by the word itself you can understand. Next one is contractibility, okay? Contractibility. That is the muscle's ability to contract. Muscles should only relax, sir. No, they should also contract. So we studied excitability, elasticity, contractibility. Who can tell the next one? So here, contractibility, they have contractile proteins, okay, who are going to do the job that we are going to study. Yes, contractile protein, elasticity, yes, extensibility, excitability, elasticity, contractibility, and we have last one is extensibility. Extensibility, okay? So you might get a little confused with elasticity and extensibility. Extensibility is also a little bit of stretching, okay, a little bit of stretching. Elasticity is even though the muscle stretches, it will be able to come back to its original position or retain its original position, okay? That is what is given exactly as a definition. You can see retain the original position. Original position, okay? So I have stretched my muscles. After that, it will be able to retain and come back to its original position. That retaining capacity, we will call it as elasticity, and extensibility, as you know, okay? Extending a little, that's all, okay? Clear? I hope it is clear. Yes, exactly. To stretch, to be able to stretch. Done and dusted. Clear? Okay? Excitability, where the muscle will be able to take the nerve impulse and react according to that, okay? Very simple. Elasticity, where it will stretch and it will be able to retain to its original position. Then contractibility, for ability of the muscle to contract, and the next is extensibility, able to do that little bit of flexibility or little bit of that stretching or extensiveness that can be done by the muscle. Don't expect a lot, okay, but very simple. Now this is clear. Chalo. Now students, on the basis of your location, okay, where it is present, we have three types of muscle which you will be studying again in depth in your animal tissues. But now I'll give you a very simple idea about it. On basis of location, okay, based on location, we have three types of muscles. skeletal muscle. Okay, skeletal muscle, cardiac muscle, and smooth muscle. Smooth muscle, or you can also call it visceral muscle. Okay, visceral because it's present in the internal organs. So I'll just give you a very little idea about it, because you will understand that very simple thing is that these things you will again study in your animal tissues. Now students, based on location means what? Where are these people present? Now if you see skeletal muscles, they are present in your, they're attached to your bones, right? They're mainly present. Skeletal means the ones who can do voluntary action, the only ones who can do voluntary action. So these are present there, and these people are voluntary. Okay, these people are voluntary. And the shape is very important. They are cylindrical, because sometimes they will give you to identify. Okay, they will give you to identify. Cylindrical. This is the simple thing. Okay. Yes. Next thing is cardiac muscle. As the name only you know, heart. Anything else do we have to remember? No ma'am. They are completely involuntary. Okay. You cannot control your heart. Can you? Then how many times you wouldn't have got depressed if you were able to control every emotions of your body, every movements of your body? Then many of us wouldn't have even fallen sick, right? So that is not possible. Heart, involuntary. Okay. Now I told cylindrical. This fellow also is cylindrical. Okay, cylindrical. Now last one is smooth muscle, or I can call it as visceral muscle also. Okay, or some places in some book, if you see visceral, you might get confused, saying that ma'am did not teach visceral. Visceral is basically because it's present internal, okay, in your stomach lining. So it's present in the visceral organs. That is why it's called as your smooth muscles. Okay, so they are present in your visceral or your internal organs. Okay, internal organs, and they are also completely involuntary. They're not in my control. Very important thing, their shape is fusiform. Okay, fusiform. Students, very easily you will be able to understand smooth muscle. They will look like this, tapering in both the ends. Okay, tapering in both the ends, like this. Okay, do you want me to draw it that side? Shall I draw the muscles, or? Yes, all of you in the chats, please let me know. I can draw the muscles. Okay, I'll draw this muscle here. I become very artistic, no? When my students have full energy, I also become an artist. The art and the artist. So students, smooth muscles will look like this. Okay, let's draw it, because I forgot to add the image. So we will just use our skills to draw the muscles. Okay. Okay, this is how smooth muscles will be. They'll have striations. Okay, they'll be completely striated, and they'll be multinucleated. Okay, like this the muscles will be, and they will also be multinucleated. I'm randomly drawing it. Okay, fine, muscle looks fine. I cannot see it because of reflection. Two same color it became, no? Chalo, no problem. Or not, actually some problems are there, because striations have to be seen clearly. So I'm using this color. Yeah, now it's clear now, students. Branching and all is there. If you want to, you know, your cardiac muscle has branching. Don't worry, structural organization, I'll teach you this. Okay, now I'm just going to give you a very slight idea about it. That's why I'm not taking much in depth. You can see that, you know, most of us, all the teachers do it in the animal tissues only, because this you have to learn detailedly there. Okay, chalo. This is how your smooth muscles will look. They'll be cylindrical. They'll have striations on them. Okay, they'll have striations on them. I think it's tic-tac. Okay, picture is okay, but you will be able to understand. The blue color one is the nucleus. They'll be multinucleated or syncytial vital. Now, next one is your heart muscle. Now, heart muscle, student, they'll be branched. Okay, they'll be branched. So, it's little hard to draw, but I'll try. Like this. Okay, they'll be branched. Different branches will be there. Right now, I'll just draw one branch. And this is how your, Okay, they'll not be multinucleated. This is how they'll be. Okay, got it all of you? Any doubts? Any doubts? Please ask me right away. No doubts, right? Will they be multinucleated or will they be single students? Are they single people or double people? Okay, they will be unstriped. Okay. Yes, tell me. Yes, they will be uninucleated, no? They will be uninucleated and it will be mostly to the center. Now, the last one is smooth muscles. Okay, smooth muscles. Smooth muscles will be fusiform shaped. Very easy to identify them because they will have tapering ends like this. Okay, they will have tapering ends. Students, cardiac muscles, some of the characteristics have actually, sometimes you can see striations, sometimes no, but majorly we will tell no striations. Sometimes we tell striations are there. So it is, we tell it usually cardiac muscle we say striated only, stripped muscle we tell because the presence is like that, right? Okay, chalo, here 1, 2, 3 and 4. Done? Any doubts? Smooth muscle, done. Branching is there. Cardiac muscle, striped. Okay, striped. Most of the time we tell striped, but it has characteristics of both. If you study detailedly about it, it has characteristics of both, striped and unstriped. Smooth, what is the question? Lie in the center, dear. No, it is uninucleated and they usually lie in the center. Okay, and smooth muscle, so 1 only, no doubts in that, they are non-striated. Okay, completely non-striated. So these are the 3 muscles. Got it? So if they give you this image and they ask you to identify, you should know you're going to identify cardiac muscle with branches, you're going to identify smooth muscles with cylindrical and multiple nucleus, and you're going to understand smooth muscle with the fusiform, this one. Okay, fusiform. Yes, it's uninucleated. Okay, fusiform. This one is uninucleated and they lie in the center. Both are uninucleated. Now we are going to come to the major part, skeletal muscle, the most beautiful part of this chapter. Okay. Yes, intercalated disc and all I'll teach you later. This is just to give you an idea about the smooth muscle and all. But we usually teach this in animal tissues, okay? Because there you will study intercalated disc, you will study why they are striped, why they are not striped, how do they have 2 characteristics, all that you will study in detail. Right now just understand that on basis of location 3 muscles are there, smooth, skeletal and cardiac muscle. We are going to talk about skeletal muscle, okay? To give you that introduction, they have just spoke about this. Chalo. Now students, look here, this is your smooth muscle. This is your skeletal muscle. And in the skeletal muscle, you will see bundles, right? You can see many bundles are there and these bundles are called as fasciculi. Now, inside the bundle, you will see these pink color structures. Those are your muscle fibers, okay? So, now just look at this image and I am going to draw the same image, okay? I am going to draw the same image and I am going to teach you. Now, look here, let's draw the muscle. So, this is how your muscle is going to look, okay? I wanted to draw a bicep, tricep, hand and all, but not possible. Okay, this is how the muscle is going to look. So, from this muscle, I'm going to enlarge this muscle. Okay, I'm going to enlarge this muscle like this and draw a structure like this. Okay, yes, this is how your muscle is going to look. Now, inside this muscle, okay, inside this muscle, there are going to be bundles. Okay, they are going to be bundles. Like this, bundles are going to be there. Let's enlarge that one bundle also. Okay, let's enlarge that one bundle also. This is the one bundle or your muscle fiber will come out. Okay, from this, inside this, okay, I've tried to draw it like this way. Let's see if we can understand. So students, this is the muscle. I have enlarged the muscle. Okay, this is the enlarged muscle, entire muscle. Now this muscle, this one right here, this one right here, like this is your muscle. Now inside this muscle there are bundles, okay? Inside this muscles there are bundles. These are the bundles that you're seeing. See, this white color structures are the bundles of muscles that you're seeing. These bundles are called as fasciculi. Okay, we'll come into that, don't worry. Now, inside this muscle bundles, each of the muscle bundles, there will be muscle fibers. Okay, muscle fibers. So, if I pull that muscle fiber out, inside that muscle fibers, your myofilament will be there. This is the flow of it. This is the flow of it. Yes, bone to muscle, that is called as tendon. So, inside this muscle bundle, let's say the one that I drew is a muscle fiber. Okay, like this muscle fiber. 1 second students. Okay, this is a muscle fiber, and this muscle fiber is what I have enlarged. This muscle fiber is what I have enlarged. Inside the muscle fiber, one muscle I have pulled out, and that muscle is going to have your myofilaments. Chalo. Now, very important thing is your entire muscle, right? Your entire muscle which I have drawn is going to be covered, okay? Is going to be covered. This covering which you are seeing, this covering which I have drawn in your orange is called as your epimysium. So your entire muscle is going to be covered with a very transparent, like it is going to look like how, you know, students, like a plastic cover, no? A plastic cover, or we could say that this kind of cover. Wait, I have an example also for you. Yeah, see this kind of a cover, no? Like this it is going to be. Like this, it's going to cover the entire muscle. That covering is called as your epimysium. I'll show you the image of that. I'll also show you the video of that. You'll be able to understand. So this orange color thing which I've drawn is called as your epimysium. Okay, it's called as your epimysium. This is the most external covering that covers the entire muscles. Okay, so I'm writing here, it covers the entire muscle. Okay, understood, ma'am. Now, there are muscle bundles. For the muscle bundles also there are coverings. Okay, for muscle bundles also they are covering. Those bundle coverings around the fascicles. Muscle bundles are called fascicles. Those are called as perimysium. Those are called as perimysium. That will cover the muscle bundle. Cover the muscle bundle. Okay, understood? So far good? So far good, yes or no? Okay, perimysium is there, that is covering the muscle bundle. Let's also label the muscle bundle. The muscle bundle, okay, muscle bundle is called fasciculi. Okay, fasciculi. That muscle bundle is called fasciculi. We will usually use the word fasciculi only. Don't think that they will use the word muscle bundle. They will use the word fasciculi only. Okay, they will use the word fasciculi only everywhere. Now students, the muscle fiber, right? Inside the muscle bundle, what is there? Muscle fibers are there. Why am I calling it fiber? Because they are long and slender. Okay, inside. So very simple. This is the muscle. Okay, inside the muscle, muscle bundles are there. Inside the muscle bundles, muscle fibers are there. And why are we calling it fiber? Because they are long and slender. Now when I pull out that one muscle fiber, see, I pulled out one muscle fiber. This muscle fiber also has a covering. Yes or no? This muscle fiber also has a covering, and this covering which I have drawn here is called as endomysium. Most innermost, no? Endo means always innermost. So endomysium, which will cover the muscle fiber. Many students get confused with muscle bundle and muscle fiber. Please understand, bundle, inside a bundle. Okay, there's a bouquet. Inside the bouquet, a lot of flowers are there. So you will call that a bouquet. Inside it, you will call it individually rose, sunflower, whatsoever. Same way, entire muscle bundle is there. Inside the muscle bundle, you will have your muscle fibers. And if I remove that muscle fiber, see, this is the muscle fiber I have removed. Inside that muscle fiber, if I pull, okay, one thing out, that will form your myofilament. Okay, that will call myofibril or myofilament. Both the words you can use. Myofibril or your myofilament. Students, a beautiful video is there for this. I will show you. Okay, I will show you very, very soon. First, try to understand this. This is most important. Now, students, to keep all the muscle bundles together. Now, let's do one thing. Let's draw this separately. Okay, let's draw this muscle, this portion. Only this portion, let's draw it separately. Here. You'll be able to understand. Okay. This is the entire, this portion. Okay. Shall I draw it oval? So you'll be able to connect easier. Okay, this is the entire, this portion that I've drawn. Now, outer covering is what? All of you in the chats, outer covering is what? By then I'll just draw the arrow mark. Okay. Okay, this is the entire thing, the outer covering. Okay, the white one which I've drawn, let's consider it as your epimysium. Okay, let's consider it as epimysium. Now, inside that, who is there? Muscle bundles are there. I'm drawing it with yellow. Okay, muscle bundles are there. Okay, like this muscle bundles are there. Okay, clear? Like this muscle bundles are there. Now, these muscle bundles are covered with what, students? All of you tell me in the chats. These muscle bundles are covered with your perimysium. Yes or no? They are covered with perimysium. Yes? Agreed, ma'am. Agreed. Very good. Very good. Many of you are answering in the chats. So, I am doing that also. I am labeling it as perimysium. Okay, perimysium. Now, what is this called? What is these yellow color circles? Those yellow color circles are the bundles, no? So, I will call those bundles as, fascicles, right? Fasciculi. Okay, done. Muscle bundle done, covering done, perimysium done. Okay, ma'am, understood. Now, inside this, ma'am, who is there? Inside this is where your muscle fibers are there, right? Inside this, muscle fibers are there. So, let's draw like this. Muscle, many will be there, students. I will not be able to draw that many, but once you see the video, you will be able to understand. This many muscle fibers will be there. This muscle fiber, why are we calling? Because they are long, okay? Long. Yes? Fine, all of you? Okay, right? Any doubts, please ask me right now. No doubts, though, we will continue. I think no doubts, right? Yes, okay, chalo. Okay, yes. Fascicles or fasciculi, okay? Plural or singular. Got it? Fascicles or your fasciculi. Clear? Sorry, students, spelling is wrong. Fascicles, okay? C-I. Fascicles, okay? Spelling is little wrong. I think now it's correct here also. When we write fast, no, sometimes we will not notice the spelling. So, you people please make spelling corrections in your, when I share the notes, okay? I think now it's correct, no? Check now. Correct, right? Fascicles. Ayyo. Fascicles. This is the spelling, okay? This is the spelling. Fascicles. Done. Okay. So, fascicles done, ma'am. Understood. The pink one is muscle fibers. Okay, that also we understood. Okay, they are called muscle fibers because they are long and slender. Okay, understood. Muscle fibers. They also have a covering, that is endomysium, which I am not going to draw because I have drawn it there clearly. Now, students, what you have to focus is that between this, okay, between this, let's say, between these muscle bundles, okay, they are all circles, no, they are all separate, they are there. To connect these muscle bundles, there is a connective tissue which is called as your fascia. Okay, like this, let's say, I think this color is not that visible. Choosing color only half of my, okay, let's keep it this way, okay. Now, students, I cannot show exactly how the connective tissue is going to be there because the diagram you will not understand, but understand to connect all the bundles together, there is a connective layer or a connective tissue layer which is called as the fasciae, okay, fascia, however you want to call. Let's connect it like this. Now, as of now, we are connecting like this, okay, like this I am going to connect it. So, see how they are connected, right? This connection is called as fascia. Fascia. This is a connective. This is a connective that will connect all the bundles together. Okay, that will connect all the bundles together. So, this diagram will help you understand the entire skeletal muscle. Let's go do a quick recap so that we know that we did not miss anything. Students, this is the muscle. Okay, students, this is the muscle. Let's say this is a person's hand. Okay, this is a person's hand. That can't be a person's hand. Okay. No students, let's not draw hands. Okay, what we can draw, we will draw. Okay, this is the muscle. So I'm writing it here, let's label it. This is the muscle. Okay. This is your skeletal muscle. Okay, skeletal muscle. And here, starts from here. So this entire muscle, okay, the entire muscle. Yes, I swear students, it's the biggest task of my life. I need one person just to choose the colors, okay, so that my notes look beautiful and my students will be able to study. I hope all of you are downloading my notes, okay. I'm using this many colors for that, otherwise everything white I would have written, yellow I would have written and finished the class. But no, I have to use colors. Okay, so this is the muscle. We have drawn an enlarged structure. Now imagine this muscle is attached to a bone. That position will be called as a tendon. Okay, muscle, bone to muscle will be called as a tendon. Now this entire muscle is there, students. This entire muscle will be covered by a covering called as your epimysium, okay, which covers the entire muscle. Then when we go slowly inside, you will be able to see muscle bundles. These muscle bundles are called as fascicles. These fascicles will be covered with a covering called as your perimysium. Okay, perimysium. This, after that, when you go inside the muscle bundle, there will be muscle fibers or your muscles, right? Muscle fibers will be there. They are called muscle fibers because they are long and slender. Okay, chalo ma'am, understood. The muscle fibers are there. When I pull out that muscle fiber, it will be long and slender like this. See, this is the muscle. Okay, this is the muscle fiber. I'm writing it here so you understand. Otherwise, you'll be like, what has this ma'am drawn? Okay, this is the muscle fiber. When I pull it out, I'll be able to get the myofilament or the myofibril, okay, which has actin and myosin, So this is the entire skeletal muscle. I hope it's clear. Any doubts? So students, now if I draw, okay, if I draw the, let's say, okay, never mind. Now you come back to this. See this, all of you. Okay, now look at this. This is your muscle. Okay, it's going a little fast, but I'll show you in replay. Okay, this is the muscle. It opens. Can you see? The entire. Can you see how the opening is happening? The epimysium is opening. Okay, okay, ma'am, understood this. Now let's look at the next one. Okay, look at the students. See, one whole thing is coming. This is what I'm going to teach you next, sarcomere. So you should be able to understand. This is your entire muscle. Now say from that one muscle fiber has come out. From that one muscle fiber, your myofibril or myofilament is coming. This will be your myosin. This will be your actin. This is going to be a Z line. Okay, this is going to be a Z line. Now I'll play the previous one. No, wait. See this once again. Muscle, muscle bundle, individual muscle cell, opening, myofibril. Okay, myofibril. Yes, clear? Clear to all of you? Chalo, let's go back to the class. So I hope that was clear to all of you. It was very easy to understand. Whole muscle, one is coming out. Okay, one thing is coming out. Just like this only. Just like this only. This is a little beautiful diagram, but that is not. Okay. Now students, from the one myofibril, did you see sarcomere? We are going to understand what is the sarcomere. Okay. Now we drew the muscle, ma'am. Okay. Now we are going to understand the sarcomere. Are you all following? Okay, chalo. Let me see your attendance, yeah. Let me see your attendance. Many of them did not mark the attendance. I'm going to be angry. Okay. Now inside the, we have pulled out the muscle fiber. Okay, this was your muscle fiber. In this muscle fiber, many myofilaments are there. Many myofilaments are there. I am pulling out, okay, one of the myofilament. In this myofilament, students, they are going to have actin and myosin. This we already know. Ma'am, myofilament, we know that actin and myosin is going to be there. So let's label this. Okay, myofilament, actin and myosin is going to be there. Now, who is this actin and myosin? They are the two proteins. Okay, they are the two contractile proteins. Contractile proteins mean the protein that is helping us for contraction. Simple, no? Contractile protein means the protein that is helping us with contraction. So we have actin, we have myosin. Who are they both? They both are the contractile protein. Now, this actin and myosin will form a region. That region is called as the sarcomere. Now, if I have to simply say what is the sarcomere, that is the functional unit of contraction. That is the exact region where contraction will happen. Okay, the exact region where the contraction will happen. So, I can call it simply the unit of a muscle fiber, or I will tell the exact functional unit of the muscle, okay, where contraction and relaxation happens. So, these two will form the contractile protein, okay, and sarcomere becomes students, last time everywhere I wrote sarcomere and none of them told. Okay, my DNA students told me, ma'am, you are writing sarcomere. So, sarcomere, okay, sarcomere is the functional unit of contraction, or functional unit of muscle, right? Functional unit of your muscle that is going to help in contraction. This is your understanding about sarcomere. Okay, is this clear? So, you have sarcomere, it's a functional unit of the muscle where contraction will happen. Now, we are going to understand further about this. Now, students, if let's take a myofilament, let's see how the whole function is going to look like. See, I am drawing it this way, okay. Obviously, this is not how it's going to look like, or I have one more way to draw. Okay, let's say we draw it like this, students, like our DNA. Okay, this one which I have drawn is your actin. Okay, this one which I have drawn is your actin. They will have this globular structure fully. Okay, they were first globular protein, then they became fibrous protein. So, this entire structure will be like this. Okay, got it? Here also same thing will continue, like this one structure is there. This is how your myofilament is going to look, okay. Then on that, let's say myosin is going to be there. Myosin is going to be a structure like this, okay, which will have the head, like this, again which will have the head like this, Let's say this is how it continues, okay, and then they will do sliding filament theory. So, this is exactly how your myofibril is going to look like. Actin chain will be there, myosin will be there, actin will be there, myosin will be there, okay. See, myofibril is this entire thing there, this whole thing, your myofibril is your entire thing. Inside that, actin and myosin is going to have a portion called as your sarcomere. Now, I will say your kidney has nephron. Nephron has two parts, glomerulus, renal tubules. So, like that, your muscle also, this is your myofibril, and inside the myofibril you have a sarcomere which will becomes the functional unit of the thing, okay, functional unit of your muscle. Is this clear to all of you? Yes, we have some more words to study, okay. Done? Fine, okay. Yes. So, they have two things, understood, ma'am. Now, students, you have to understand this. I will not call plasma membrane as plasma membrane. I will call it what? Sarcolemma. Okay, I will call it sarcolemma. So, this will be the plasma membrane. Here one plasma membrane will be, this will be called as sarcolemma. So, I am going to write it here, okay. Plasma membrane of your muscle, okay. Plasma membrane of your muscle will be called as sarcolemma. Yeah, sarcolemma. Any doubts? Cytoplasm will be called as sarcoplasm. Okay, cytoplasm will be called as sarcoplasm. Endoplasmic reticulum will be called as sarcoplasmic reticulum. Okay, these people are very special. They are like, I don't want your normal names. I'll use my own names. So, it will be called as sarcoplasmic reticulum. Sarcoplasmic reticulum. So, very important thing about your sarcoplasmic reticulum is they are going to store calcium. Okay, they are going to store calcium ions. Why this is going to be important? Because when we are going to do actin myosin marriage, this fellow is going to be the most important fellow. Okay, without calcium, none of the marriage is going to happen. So this fellow is going to help. So plasma membrane will be called as sarcolemma. So in your textbook, later on, okay, now in the stages they'll introduce you. Later on, they'll only use the word sarcolemma, sarcoplasmic reticulum, sarcoplasm. So you should be understanding. Thank you. Again, I don't know why I'm very used to writing this, I feel. Okay, sarcoplasm, sarcolemma. Okay, sarcoplasm, sarcolemma. Understood? Okay, got it? So it's very clear about this. Now, imagine you just have to know one, let's say one calculation you have to know. How many, one thick filament. Now, please understand, myosin, actin is there. Okay, you told about myosin and actin, ma'am. This myosin, no students, is the thick filament. Myosin is a thick filament. What is it? Thick filament. Now, why are we telling thick and thin? Because you should understand your skeletal muscle is striated. They have stripes, right? They have stripes on them. So, for them to have the stripes, they should have a thick filament, thin filament, or form dark bands and light bands. Got it? Whenever you have to form a striation in the muscle, what do I mean by striation? Like this, stripes should be there. No? Some places will be light, some places will be dark, some places will be light, some places it will be dark. For that striation to happen, you have to have thick bands and thin bands, or dark bands and light bands, right? Who is going to contribute for that thick band and thin band? Is going to be myosin, which is the thick filament, and actin, okay, actin going to be the thin filament. Okay, actin going to be the thin filament. Now students, the way to remember this is actin, tin, 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 tin. Okay, actin, tin, 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 tin. So you repeat actin 2 times, it will become thin. Okay, so actin is thin filament, myosin is thick filament. Got it, all of you? Got it, all of you? Any doubts so far? Yes, no doubts? Okay, no doubts, right? Okay, fine. Now actin is actually a 6-ring structure. Okay, if I have to draw actin, it will be a 6-ring structure. Let's say like this. Now students, how much chemistry will I teach? Okay, it's going to look like this. Okay, one 6-ring structure it is going to be actin. So here, 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 here. So this is just to understand how many thick filaments, how many thin filaments. That's all. Okay. Yes, tin, tin, tin. Okay, that's how you will tell. Very good, very good. Okay. So now students, one thin filament. Okay, one thin filament will be surrounded by how many thick filaments is the question. One thin filament means actin will be covered by how many thick filaments. So if I draw myosin, let's take myosin is going to be a triangular shape. Okay, myosin is going to be a triangular shape. This is one thin filament. If I have to cover it, I have to draw. I have to learn too much of math, students. Okay, lot of maths I have to draw. So let's say I'm covering this. This is easier. See, this is how it will be. Okay, one whole actin is there. This is myosin. Myosin will be 3, actin will be very good, very good. Okay, myosin will be a 3-ring structure, actin will be a 6-ring structure. So one thin filament. This is one thin filament. One thin filament will be covered by 3 thick filament. That's all. That's all you have to remember. Okay, so here I am writing one thin filament. One thin filament will be covered by 3 thick filaments. Three thick filaments. Okay, that's all. Clear? Yes or no? So obviously one thick filament will be covered by 6 thin filaments. Yes or no? Now if I say put this, let's take this only 1, 2, 3, 4, 5, 6. So one thick filament by 6 thin filament, one thin filament by 3 thick filaments. That's all. Okay, that's all. So both also in this diagram only we'll be able to represent. So I'm writing it here, students. One thick filament will be covered by 6 thin filaments. This is just a number. If you can, try to learn it. If you can't, it's okay. Done? Okay, sorted. This is sorted. Okay. Now, I should have wrote it here. Never mind. Okay, now we are going to learn the sarcomere. Okay. I hope I haven't put one more slide. Okay, yeah. Students, very, very important part. How does this actual proteins contract? That is what we are going to learn. Okay. So, all of you look here. We are going to draw two structures. Let's take two colors. Okay. We will fix the colors. I am going to draw myosin with orange. Okay. I am going to draw myosin with orange. Let's draw it like this. Oh, wow. I told orange and yellow came. No obedience only board has. Okay. This is your myosin. This is your myosin. Very important diagram. Okay. Once we draw it, you will understand it. How contraction exactly happens. Okay. Okay. I have drawn a longer one just to make it more clear to all of you. And let's take yellow for your actin. Okay. Let's take yellow for your actin. Okay. Actin. Actin. Okay. Again actin and actin. Okay, students, done? Yes. So, this is how the filament, okay, how it's going to be looking like. Now, in that, I am going to draw some lines as well, okay. I am going to draw one Z line. What am I going to draw? One Z line. Actually, if you see it, it will connect like this, but that is too difficult to make you understand. So, I am going to draw it right in the center itself, okay. When we go further, we will learn that way. I am going to draw one in the center. Here also I am going to draw one. Okay, I am going to draw one. Okay, ma'am, understood. This is clear? Chalo. Now, between the M, okay, this is myosin, no? Between the M, I am going to draw one M line also. I am going to draw one M line as well. Very easy it is going to be. Don't take tension. We will learn it very simple. Okay, this is the M line. Okay, this is the M line. Now, let's start labeling what is what. This is how a sarcomere is going to look like. Okay, this is an entire sarcomere. All of you remember the GIF I showed you. Now, let's go back to the GIF and look at the next one. Look at this one, students. Where's the remote? Yeah. All of you see this? Can you see how it's coming out? This is what I've drawn. Okay, this is what I've drawn. So, from the myofibril, this is the whole thing. From that, you can see one sarcomere coming out. Right. This sarcomere is what I've drawn. The orange one is your myosin, the blue one is your actin, and this one which you see is the Z line. Can you see all of you? Fine? Done? Okay? Chalo. Let's go back to the class. So, this is what I've drawn, okay? Here we have one lines. Let's label what we have written. The yellow one is your actin. Okay? Actin. Actin. This zigzag line is your Z line. Your zigzag line is your Z line. This orange one is your myosin. Okay. Orange one is your myosin, which is your thick filament. Now, what is going to happen, ma'am? What are we going to understand? First, we will do one thing. We will write few bands. Okay. We will write few bands. After that, you will be able to understand how does contraction happens. Okay. Now, students, the band, okay, first band is going to be a band where only actin is present. A band where only actin is present. Since only actin is present, I will call that band as isotropic band. Iso word means same, right? Iso means always same. So, a band where only actin is present, I am going to call it the I band. What band? I band. We will write the full form later. Right now, try to only understand this. The band which has only actin, I band. Okay, ma'am. See, if I look into the center, no, actin also is there, myosin also is there. It's not separate. It's not separate. They are little mixed up. So, this area, this portion, I will call it the A band or anisotropic band. What is it called? The A band or the anisotropic band. Iso means same, aniso means different. So, iso means actin, aniso means here different. Mixing has happened. So, anisotropic band. Okay, ma'am, understood. I band, this will also be called as the I band. Okay, this is also called the I band. Clear? Anyone wants to write? Who is this? I am your friend, yaar. Guess my name. Please guess the name, students. Here, more than NEET exam, guessing is happening. Okay, please guess the name. Very important for him. Don't disappoint him. Fast, fast. Guess the name and finish it off. Chalo, ready? Okay. Okay, yes. Now, ma'am, I understood I band is isotropic band where only actin is present. A band is anisotropic band where actin and myosin is present. Again, I band is there. Okay, ma'am, Z line I understood, the line that will hold the actin. M line I understood, the line that is going to hold the myosin. This much you understood. I can neither call this one as light band, dark band. So, always think of the muscle. Always think of the muscle. That muscle will have a dark striation and a light striation. Okay, light striation. Now, I band is be the one where has light band, because only actin is there. Thin one, no, thin fellow is there. Here thick fellow is there. So here thick and thin is there, so it will be the dark band. This will be again the light band. Now, very important things, very important things. The portion, okay, in the middle there is a portion where only myosin is present. All of you listen to me carefully. The portion where only myosin is present here, okay, this portion I will call it H zone. What zone, students? H zone. Okay, here I am writing this portion is called the H zone. The portion where only myosin is present. The portion where only myosin is present will be called as the H zone. Okay. Now, here, if I look here, if I look here, the portion where actin and myosin are overlapping. Let's say this is the myosin. Okay, this is the actin. The portion where both of them are overlapping, I will call it the overlapping zone or the O zone. Okay, O zone, students, O band also you can say. Okay, O band also you can say. This portion, this is this one is this portion. This portion where overlapping is happening. Can you see? Myosin is there, actin is there. Myosin is there, actin is there. This portion will be called as the overlapping portion or the O zone. Okay, O zone. Got it? Done? Done? H zone done, O zone done, Z line done, I band done, A band done, M line done, all of this is done. Now, the question is, when a contraction happens, who is going to make the movement? When contraction happens, who is going to make the movement? All of you listen here. When contraction is going to happen, students, the Z line is going to move forward. Okay? When contraction happens, the Z line is going to move forward. This way contraction will happen. So many zones will become less, many zones will become big, all that we will understand. But understand, whenever contraction happens, the Z line will always move forward because actin is going to slide on the myosin. Myosin is not going to slide on the actin. Actin is going to slide on the myosin. Got it? So here the Z line, which is holding the thin filament, is going to push forward, move forward. Now I told you sarcomere is the structural and the, let's say, functional unit of the muscle. What is a sarcomere? Sarcomere starts from the first Z line to the next Z line. That means between these two, this Z line to this Z line is a sarcomere. Is a sarcomere. All of you understood? What is a sarcomere? Starts from one Z line to another Z line. Then from this Z line to this Z line, next sarcomere. From this Z line to this Z line, next sarcomere. So sarcomeres are between two, okay, sarcomeres are between two consecutive Z lines, right? Are between two Z lines. Understood? Yes or no? Yes? Okay, understood. Now, very important thing. All of you just look here and tell me, one sarcomere will have how many bands? I band, A band, what is going to happen? Okay, what is going to happen? See students, one sarcomere, let's say these two are the sarcomeres, right? If you look here exactly, you will see half I band, one full A band. Agreed or disagreed? Half I band, okay, half I band, full A band, half I band. So I would say one sarcomere, okay, one sarcomere constitutes half I band, okay, plus one more half, right, plus one more half I band, plus one A band, okay, plus one A band. So simple formula, I always like making formulas easy because maths does not love me, is that I will write one I band plus one A band forms a sarcomere. So one sarcomere will have one I band, one A band. Yes or no? Simple maths, half plus half I did, okay? Simple maths, half plus half I did. Clear to all of you? Done, students? Okay? Yo, this has become one issue only now. I have to go to class to make this issue rectified. Sometimes you people can adjust, okay? Sometimes if I'm free, I'll edit like this, okay? Yes. Done? Done. Okay, so students, so very simple. If contraction has to happen, my actin and myosin have to do a theory called as sliding filament theory, right? So that contraction to happen, I need a sarcomere, right? I need a sarcomere. Sarcomere is between both the Z lines and they are going to push forward. Now, when they push forward, okay, when they push forward, let's see what are the things that will reduce and what are the things that will increase. Are you all ready for it? Yes, all of you drink some water. All of you drink some water. Okay, and mark your attendance. Jaldi, jaldi. Right, ma'am needs some energy. I'm hungry. One more class, Kamali. Dr. Stephen is telling funny things. Okay, is this clear? Is this clear? Okay, fine. Now we are going to understand if a muscle contraction happens. Okay, let's say muscle has to push forward. Okay, then what are the things that will reduce? Before that, I'm going to write some full forms for all of you to help you because I've written only head zone, eye zone. No, students who are studying this chapter for the first time, they might find it very difficult. So actin is going to be the thin filaments. So I band, students, all of you write with me. I band is going to be isotropic band. Okay, isotropic band, which is also I can call it as the light band. Okay, light band. Next is A band. A band, which is anisotropic band. Anisotropic band, which can be called as the dark band. This people only will cause the striations, okay? Then we have Z line, M line, no full form for that. Then we have what, students? A band, I band, O zone. Don't write your O zone, okay? O zone called as your overlapping zone. Okay, overlapping zone. O band also you can tell, no problem. Then you have head zone, which is called as Henson zone, where only myosin is present. Okay, Henson zone, where only myosin is present. Did we miss something? Head zone, O zone, I, A, done, no? That's all right? Yes. Clear? Students, where you will find a better diagram? Okay, so definitely comment and only go and share the video with as much as friends possible. Okay, see how nicely I'm trying to make you understand. Students, we didn't label M line, yeah? M line. Okay, M line. Done. Okay, this is clear. Now, if contraction happens, before that I'll show you one image of it. Now this one, next image. Okay, next image, just look at it. You'll understand how contraction happens. See, look here students, how contraction is happening. Can you see? So this is the Z line. Okay, this is your actin. Kuch is there. This is your myosin, and these are the heads of your myosin. Okay, can you see how they're moving forward? See, they're moving forward, they're going back. They're moving forward, they're going back. Can you see this, all of you? Okay, this is how contraction is going to happen. So you can see that these people are coming closer, but you can see actin is the one who is making the effort. Myosin is not doing any effort. Okay, myosin is not doing any effort. You people can decide who is the boy, who is the girl. Okay, so here you can see they're pushing and they're doing contraction. Got it? Now we have to know which are the zones which are reducing, which are the zones that are increasing. Okay, thank you so much, Asim. Thank you so much. Okay, chalo, let's go back. Let's go back. Okay, this is done. Okay, now we will I want to add that. I think I did it in the last. Chalo, we'll write it here, students. Who is decreasing, who is increasing? You just tell me. According to your thought process, you can tell me, ma'am, maybe this will be increasing, maybe this will be decreasing. I'll correct you after that. Okay, I'll correct you after that. Who will be increasing, who will be decreasing? No friends, ma'am. People are telling such sad stories every day. Whenever I tell you to share, you'll be like, ma'am, no friends, ma'am. You are only my only friend. You are the only person that I talk to, see, I look into, I wait for your classes. You are my only friend. They don't want to share. They just want to tell that, ma'am, you are my friend. People know nicely you need hittings. Okay, all what is that? Ulta pulta work you people have in your hand. Okay, all ulta pulta works. One person is not there who will tell, ma'am, yes, ma'am, yeah, what you tell is true, ma'am, we will share it with the friends. Nah, never. Okay, shall we continue? After this, we have, students, I forgot to add that image in your textbook, but it's okay, I will do it. Okay, I'll do it. So now we'll see how does the muscle contraction, if the muscle contracts, who are the people who are going to decrease, who are the people who are going to increase. Okay, very important. In your textbook, last you can see one image where they show like lines and they show like this zigzag. No, now with that only I'm going to explain you. So when a contraction happens, okay, when contraction, both started acting. Okay, when contraction happens, when contraction happens, first of all you have to understand one thing. This A band, no, the dark band, he'll be like, I will not move. If you want, you move. So first person who is going to be constant, okay, is going to be your A band. A band is going to be constant. Oh no. Okay, constant. Students, this board will collapse at any time. A band is constant. Then everyone try to think and tell me what would be constant, what would be this. See, very simple, students. I band is the one who is coming forward. Very simple, no? Okay. So I band is the one who is moving forward. Yes or no? So obviously I band is going to decrease in size, no? I band is going to decrease. Head zone also is going to decrease. Yes or no? Where myosin is there, no? There the actin also will come off. So head zone also will decrease. Head zone also will decrease. The only person who is going to increase is your O zone. O zone is going to increase. Why it will increase? Very simple thing. Where, see, myosin is there, actin was here before. When contraction happen, actin will push forward. So more overlapping zones, right? More overlapping zones will be there. So, okay, more overlapping zones. So O zone will increase. M line also is going to be constant. Okay, M line is going to be constant. Got it, all of you? Any doubts? No doubts, right? Okay, Z line will move forward. Understand? Z line is the first person. It will move forward. So from this we can understand one thing, that myosin is not doing much efforts. Actin is the one who is doing the effort. Okay, don't write this in the exam. They will come and meet me and they'll be like, who is the teacher who taught you? Okay, this is just to make you remember. Myosin is actually very less effort person because M line also is constant. If you see A line or A band also is constant. If you see otherwise, I band is the one making the effort, Z line is the one who is making the effort. So actin is the one who is going to do the sliding. Okay, actin is the one who is going to do the sliding. Actin is the one who is going to do all the sliding. Done, then are done. Okay, chalo. Now we are going to get into one marriage invitation of actin and myosin. Okay, first we will start with myosin, then we will slowly go to actin. Okay, students, drink water. Drink water, very, very important. Okay. Should I involve in your conversation? Should I involve in your conversation? You're my bestie, I am your bestie, we are besties. All the besties tomorrow, next year NEET exam you have to write. Okay, that time all the besties will look at each other and sit. Hey, what was ma'am telling? Thick or thin filament? We were playing bestie, bestie, we forgot. Okay, you come back and cry, then I'll give you one. Okay, so now students, myosin, we all know it is the thick filament. Okay, thick filament. Thick filament. This myosin is made up of a monomer. Okay, it's made up of, the monomer is called as meromyosin. Monomer is called as meromyosin. Got it? Now we are going to draw the structure of myosin. After that, I'll explain you the rest. Chick, 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 chick. Now, like your DNA only we will draw. Okay. Okay. This is how your myosin is going. Oh no, two became, both should be equal size, students. I will try my best to draw. I'll draw smaller head, so it will be easy for me to continue. Yes. Okay. This is your who? This is your myosin. Okay, this is your myosin. Can you see? Okay, yes. 160. Suhas, you have to practice, you have to find where you are making mistake. Okay, is it assertion reasoning? Is it some topic specifically? You have to find out. Now students, look here. You look like this is a snake and two heads are there, like your cobra, no? Same way your myosin is there. It has a globular head. It has a short short arm and a tail. Okay, let's call it like this. So this person, this one is called the tail. This is the tail. Tail. Okay, ma'am, tail, understood. Okay. This portion is called the short arm. We'll tell it cross arm also. This whole portion cross arm. This will be the short arm. Okay, short arm. And this is going to be a globular head. Okay, globular head. This is what myosin has. Myosin has. Okay. Tail it has, a short arm it has, a head it has. You can imagine it look like a person. Okay. Tail is there, neck is there and head is there. Same way, tail is there, short arm is there and globular head is there. Now, the most important part is depending on their density. Basis of density, very simple. Tail is going to be LMM, that is light meromyosin. Okay, light meromyosin, depending on the density. Okay, short arm and head together. Okay, short arm and head together is going to be heavy meromyosin. Students, PYQs have come on this. So, be careful, do not skip this. LMM means light meromyosin, HMM means heavy meromyosin based on density. Simply based on density. Don't try to, understand more about this. That's not required for you. Okay, you just have to know who is light and who is heavy. Okay, light meromyosin and HMM is heavy meromyosin. Okay, heavy meromyosin. That's all. Okay, HMM, LMM. Clear? Now, Very important. Two villains are sitting on your, one villain is sitting on your myosin's head. That is your ATP binding site. What is it? ATP binding site. Very, very important. Here, this is where the story actually starts. All this was just introduction, okay? Now we are going to come to climax and to the tragedy that is going to happen. These two places that is there, that is your ATP binding site. What is it? ATP binding site, okay? On the top of that, we have an actin binding site. We have who? Actin binding site. Just now I told you, if the sarcomere have to do contraction, your actin has to slide over myosin. For actin to slide over myosin, something should happen, no? For actin and myosin should meet. But meeting is problematic because these two people are not going to, who is not going to, ATP is not going to allow meeting of them. So, ATP binding site is the first villain that I have introduced into the story. ATP binding site. Students, actually they are very power people, okay? But for our understanding, we are making them villain, okay? Because if they are not there, then muscle will always be contracted only. So, ATP binding site, and this is going to be your actin binding site. This is going to be actin binding site. This binding site will only be visible when this ATP goes out of this myosin's head. Okay, this actin binding site will only be visible when ATP goes out. Got it, all of you? Or got it? Chalo. This is your myosin. Any doubts in this, students? Any doubts? Ask me right now. After that, don't cry and tell later. Okay. Now, this Myromyosin, now I have drawn one chain, no? This Myromyosin, if you want to know the exact structure, has a 6 polypeptide chain. Okay, it has a 6 polypeptide chain structure. 6 polypeptide chain. Now, looking at it, what do you understand? It will have 2 long chains. These are the 2 long chains. 2 long chains. And obviously 6 minus 2 is 4, 4 short chains. Ma'am, but you did not draw short chain only. Where do you think is the short chain, students? The short chain, where do you think the short chain is going to be? On the head only, okay, on the head. This is the short chain. Like this, like this, like this, like this. The white color which I have drawn is the short chain. These 2 are the long polypeptide chain. So, 6 polypeptide chain this Myromyosin will have, or your myosin as such. They will have 2 long chains, 4 short chains. These are the 4 short chains which you will see on the head, and these are the 2 long chains. Clear? Clear or not? Any doubts? I don't think any doubts will be there. Okay, chalo. Let's continue. Okay, let's continue. So, this is the globular head, this is the short arm. So, when you are writing HMM, which is the heavy Myromyosin, you should include the short arm and head. Don't forget that, okay, short arm and head. So, this is the chain of myosin. Now, you understood, no? One villain that is there is the ATP binding site. If ATP is there, actin binding site will not be visible. If actin binding site is not visible, then the actin will not be able to come and bind with the myosin. If actin does not bind with the myosin, contraction of the muscle won't happen. This is the story, okay, this is the story. Now, let's see what is the problem in actin structure. What is the problem in actin structure? So, students, let's take first color, okay. So, actin was first a globular protein, okay. Actin was first a globular protein like this, okay. We will call it G protein. First it was called as G protein, globular protein, and then it became F protein. F protein means fibrous protein, okay, fibrous protein. We will draw one infinity sign like N structure. This is just for our easy understanding. It's called as F actin, okay. G actin, okay, I'll write it G actin, not only protein, okay, G actin and F actin. F actin means fibrous actin, okay. Now, on this, this structure will be there, okay. Wait, let's do one thing to make it easier. We'll put dots. Perfect, okay, perfect. So, these globular proteins arrange themselves to form a fibrous actin, okay, fibrous actin. Now comes the entry of villains. This is the F actin structure. This F actin structure also has a myosin binding site, but it will not be visible to any of you. Why? Because there are 2 villains in actin, that is tropomyosin and troponin, okay. Now, students, let's first draw troponin. Okay, let's first draw troponin. Troponin will be present at regular intervals. Okay, now for our understanding, we are drawing it like this. Okay, this is how similarly it will be. Okay, regular intervals 1, 2 and 3. Okay, 1, 2, 3 I've put here. 1, 2, 3. Center also I'm putting 1, 2, 3. Okay, this is your troponin. Now one more villain I told, no? That villain also will draw. That villain is going to come through the center. Okay, like this. Like this. This is the structure of your actin. Are you able to see? Okay, are you able to see the dots? I think the dots are not that visible. Wait students, let me use, I have better colors. Okay, why am I even struggling? Okay, wait, I'm making it bigger. Now I think it should be visible. 1, 2, 3. Visible, no? Ha, done. Now this troponin and tropomyosin we are going to draw. Let's draw this. Okay, this is, wait, too small. Okay, this is the troponin. Okay, troponin and your tropomyosin. Let's first label it. This is your F actin, students. This is your troponin. Okay, troponin has some subunits and all which we will understand. I'm just writing now troponin. This is your tropomyosin. Myosin. These are the two villains on the actin. Now these villains are what they are going to do, no? These villains are not going to show the myosin binding site. Students, behind this myosin binding site is there, but it will not be visible. It will not be visible because of the troponin and tropomyosin. Now let's see who is this problem. Okay, this is the three structures of your troponin. Three circles are there. This is going to be called as the TPI. TPI. Okay, this is TPI. Okay, this is TP. This is TPC. This is TPC. Now, what is this TPI and all this? I will just tell you about this. This is the inhibitor. That means this is the person who is going to stop actin and myosin binding. Okay, TPC is the one who will bind with the calcium. Okay, who will bind with the calcium. Then this is the troponin by itself, who is going to stop. Okay, TPC, yes, TPT. Okay, this one, T I didn't write, no? Yeah. This is the troponin by itself, who is going to inhibit actin and myosin to bind. This is the structure. This is the structure. Clear? Okay. Has the board not applied? Huh. Okay. Clear, students? TPC, TPT, TPI. TPI is the inhibitor, TPT is the troponin, TPC is the one which will bind the calcium. Now, this fellow is going to come for help for us. Okay, I'll tell you why. So, here what has happened, here the myosin binding site is not visible. Okay, myosin binding site. Okay, myosin. Students, you will get this note on Vedantu NEET English Telegram. So, use this notes to study. Myosin binding site will be masked. Will be masked. So, this way contraction will not happen. Contraction will not happen. Now, what's our job? All of you tell me in the chats, what's our job? Our main job is to make sure that these two people meet and contraction of muscles happen. Okay, contraction of muscles happen. Before that, you have to know few things. The few things that you have to know is this troponin and tropomyosin. Okay, troponin and tropomyosin, they are regulatory proteins. Those people were contractile protein, these people are regulatory proteins which will control the binding. Okay, control the action. Done, done? Okay, ma'am, done. Okay, okay, done. This one. Now, students, look at the structure given in your NCERT. See, this is how it is. Can you see that thread like a rope it is going? No, that is your tropomyosin, and this one is your troponin. So, you can see here troponin, tropomyosin and F actin. Okay, fibrous actin. So, I have drawn it like infinity sign for my easy way. But if you see, they are like 2-3 ropes going around. Okay, now look at myosin. It looks like a cute snake, no? One, two small head is there, like a cute snake, and it has an ATP binding site, head, cross arm. This whole thing is a cross arm. This portion is a short arm. Okay, short arm. This whole thing is a cross arm, head, ATP, actin binding site. This is what I taught you. Image have to be clear in your head because this image will be asked in your exam. Okay, this is the image that they will ask. Done? Okay, done. Now students, marriage is going to happen. Whose marriage? Actins and Myosin's marriage is going to happen. That marriage will be called as cross bridge formation, okay? I always call it a marriage because it's easy for me to understand, okay? So here what we are going to do is we are going to be the brokers. So we and all of us are going to be the brokers who are going to make sure that this marriage happen. Now ma'am, what are you trying to say? How can we make the marriage happen? Very simple. If a nerve impulse is coming, my muscle have the ability to get excited. The nerve impulse has told that, okay, let's say Madhumati to retrieve her hand. The signal has come from the CNS, it has carried by the motor neuron and it has come to the, let's say the neuromuscular junction, where the neurons and the muscles, okay, will be like, this is the muscle, neurons will be like this. That is where our broker job is. That means a signal has come. Now because of the signal, the muscle has to contract, and immediately the contractile proteins, both of them will make their actin binding side, myosin binding side visible, and they will try to bind with each other. This is what you're going to understand. Now look here. Neuromuscular junction, student, this is the motor neuron, okay? It is bringing the signal, and here you can see this is your muscle. Can you see the muscle? Can you see the actin, myosin and all of that? Can you see the muscle? Now these people are going to bring the impulse, and in that impulse what is going to be there? Okay, what is going to be there? They are going to have your acetylcholine, neurotransmitter. That neurotransmitter is going to bind, and the muscle immediately actin, myosin. Okay, now students, wait, I will teach you. Okay, look here, this is the muscle, this is the diagram. First I will teach you, then you will come back to this diagram. Okay, chalo. Neuromuscular junction. I am going to draw it like one box and teach you, because otherwise be too difficult to, diagrammatic representation will be difficult. Okay, this is the, not the box. Okay, this is the box, but there's a problem with the box. Problem is that I don't want that portion. Can I erase something? Okay, students, 1 second. I have to only draw because I cannot. Yeah, chalo. Never mind. Okay, this is the neuromuscular junction. Okay, they'll have something called as the T tubules. These are the T tubules. Okay. So this is a junction between your muscles. Okay, thank you, Soni. This is a junction between your muscles and your nerves. Okay, motor neuron. Now imagine motor neuron is bringing the signal. Okay, motor neuron is bringing the signal. Okay, motor neuron is bringing the signal like this. In this, neurotransmitters there, no? All of you know, neurotransmitters will be there. They will break open, right? They will break open and they will release acetylcholine. Now, before that, understand, on the muscles, there are going to be receptors. Because they are excitable, we already know they were going to have receptors, right? They are going to have receptors. So, those receptors will be waiting to collect the neurotransmitter that is going to come. Okay, I am drawing like one cup, okay? Like this I am drawing. Receptors. These are the receptors. Now this acetylcholine is going to break open. This acetylcholine is going to break open and they are going to be collected by these receptors. Okay, these receptors are going to collect the nerve impulse that has come with the help of a neurotransmitter. Okay ma'am, I understood this neural control and coordination. I know that okay, nerve impulse will come and action potential will be created. As soon as this motor neuron bought the signal, an action potential is going to be created. Okay ma'am, I know this. Now this action potential got created, because of which, okay, I am writing it here. Okay, here an action potential is created. Okay, action potential, okay, is going to happen. Okay, is created, because of which whenever an action potential happens, there is an influx of sodium. All of you know this, right? So students, here what is going to happen, all of you listen here carefully. Sodium, okay, sodium is going to influx through this T tubules. These are the T tubules. Okay, even if you don't care about, I mean, even if you forget the tubule is fine, just know that sodium influx will happen. Okay, Na plus will keep coming in. This you will study again in neural control and coordination. This influx will keep happening because the action potential got created. Full sodium influx everywhere, everywhere sodium influx is going to happen. Because the action potential got created. The motor neuron bought the signal from the brain telling that retrieve the hand. Now impulse have started coming, because of which action potential is happening, and there is an influx of sodium. Now ma'am, why are you telling about the influx of sodium? You will understand here, because students, here inside this your endoplasmic reticulum is going to be there. Who is going to be there? Endoplasmic reticulum is going to be there, which is called as the sarcoplasmic reticulum. Inside the sarcoplasmic reticulum, who was there, students? Calcium was there. Yes or no? Calcium storage house these people were. C8 plus. Okay. Calcium was there, no? They are the storehouse of calcium. Calcium. Now, when sodium influx happen, it will allow the calcium to get released from the sarcoplasmic reticulum. Got it? Because impulse have come, muscle has got excited, muscle knows that the muscle has to retrieve the hand, because Madhumati's hand has touched a hot object. So, retrieving is pakka. Now, what you have to understand is, when influx of sodium happens, that means when an action potential is created, influx of sodium will happen, and because of which this calcium will get released. Okay, this calcium will get released. And students, this calcium release is not going to be a small thing. Why? Because on the actin, if you all remember, there was a troponin, right? There was a TPC, right? One among this in the actin, right? Here also, let's draw one more. Okay, this is your actin structure. Okay, this is your actin structure. In the actin, there was a TPC structure, right? TPC. This TPC is going to get conformation. Okay, this is TPI. This is TPC. Students, this TPC is going to take this calcium and do a conformation. That means, let's say simple words, okay, in a very funny way, I'll explain it to you. Very simple thing. This fellow, okay, this fellow will get distracted because of calcium. Okay, very simple. Troponin will get distracted because of calcium. Okay, because of calcium. And because of this, what will happen? Automatically myosin binding site will start getting visible. So the first job of us as brokers was to send signal. We send the signal. Yes ma'am, we send the signal, and action potential was created. After the action potential was created, the second thing that happened was influx of sodium happened, right? Influx of sodium happened. This influx of sodium led to the third step, that is release of calcium. Release of calcium from sarcoplasmic. Release of calcium from sarcoplasmic reticulum. Yes or no? All of you, are you getting this? If you don't understand this, you will not understand sliding filament theory. Okay, release of calcium from sarcoplasmic reticulum. Okay, now this release has confronted, okay, conformation, okay, conformation of troponin has happened. Okay, troponin has happened, which will lead to the tropomyosin also getting distracted. So one person will distract, everyone will get distracted. Okay, so this will allow conformation, and this will lead to what, students? The most important thing that we waited for, that is myosin binding site will be unmasked. Myosin binding site, okay, will be unmasked. Yes or no? Clear or not? The most exciting part is that the myosin binding site has to be visible. Now myosin binding site is visible. Okay, I was conscious. This time I was conscious. I was like, this people will tell, so better I will write it properly. Yes? So students, let's call actin as the bride. Okay, so now the bride is ready. Okay, makeup artist came, everything happened, distraction happened, both the villains also got removed. Now actin is completely ready. Okay, actin is ready for Shadi, Kalyanam, right? So actin is ready for marriage. Marriage is cross bridge formation. Ready or not? Actin is ready. Now we have to make myosin ready. Only actin say what we will do. Actin and myosin should be there, no? Then only muscle contraction will happen. So is this clear to all of you? Any doubts? Any doubts? Okay, this is the motor nerve that is bringing signal, and this is, students, this is ACH, okay, acetylcholine. This dot dot which I have drawn, no, is your ACH, okay, acetylcholine. These are your receptors. I am trying to label as much as possible. This is your receptor and this is your neuromuscular junction. Okay, you can call it as neurotubular junction also, because these T tubules are there. These are the T tubules. Okay, these are the T tubules. Now go back to the diagram. Tell me if you are able to understand this. So students, what is happening? This is a motor neuron which is bringing the neurotransmitter, and this is, you can see, motor end plate, and from here what is going to happen? They will release this, and because of which actin's myosin binding site will be visible. Now look here all of you, this is the muscle, okay ma'am, acetylcholine got released, can you see? And here can you see automatically actin, this is the actin. Can you see these dots, no, blue color dots here? These are the acetylcholine. Now they will go and they will release the, here acetylcholine, they will go, and because of which calcium will get released and the actin's site will be visible, and automatically ATP hydrolysis will happen and they both will start binding. I will show you that also. Okay, I'll show you that. Okay, look here all of you. See, those are the calciums. They have bound with the troponin, because of which myosin binding site will be visible. Now see, myosin is attached. Can you see? Now look here. Here acetylcholine has come. Okay. Now see, they will open. Impulse have come. Receptors took it. Now they will send signal, and automatically calcium release will happen. They will go and bind with troponin, and automatically myosin binding site will be visible. Myosin will start binding. Then sliding will happen. Okay. See, movement will happen. Clear or no? Check, check. All of you see for 2 minutes. After that, I'll continue. I want some fire emojis in the chat if you understood, and those who haven't marked the attendance, mark your attendance. Okay? I know you're all hungry, even I'm hungry. So let's finish it. Only sliding filament theory and some MCQs are there. After that, I'll meet you at 4 o'clock. Clear? This is clear, no? Very clear? Okay, so here you can see binding sites, automatically myosin is binding. That is the myosin head. Okay, myosin head. Okay. See here, impulse came, receptors took it, signal happened, calcium got released, they went and bound. Automatically you can see actin got twisted. That is to show the myosin binding site, and this myosin head will go and attached. Okay, go and attach. I think next one also is like that. Yeah, here, okay, this is sliding filament theory. I'll show you now. Okay, done students? Okay, this is the diagram. Okay, now next is sliding filament theory. Okay, sliding filament theory, first I'll explain you with our NCERT diagram, then I'll explain you with that other diagram. Chalo. Okay. Okay, so students, look here. Sliding filament theory means first we made the actin ready. Okay, actin is ready. Now our next job is to make sure that the myosin also is ready. On myosin, tell me who is the villain who is sitting? ATP binding site. Now ATP binding site has ATP. That ATP has to leave the myosin head for the actin to see the actin binding site. So all of you look here. The first job is to hydrolyze ATP. Okay, the first ever job for us is to hydrolyze ATP. That means break down ATP. And do understand the myosin itself is a ATPase active. That means it has the ability to break down ATP. Okay. So hydrolysis of ATP will happen. That will form ADP plus PI. The moment hydrolysis of ATP happen, the actin binding site will be visible on the myosin. Okay, actin binding site will be visible on the myosin. Actin is already can show the myosin binding site. So now they will go and form a cross bridge. They'll form cross bridge. This is the marriage. Okay, this is the marriage we were talking about. Cross bridge has to be formed. Okay, ma'am, cross bridge is formed. Now, formation of cross bridge, understood. Now, here, what is going to happen, students? This ADP and PI is on myosin's head. This ADP and PI cannot stay for longer inside the myosin head. So, what they will do? They will push themselves outside. They will push themselves outside, and that will cause a power stroke. That will cause a power stroke. Got it? That will cause a power stroke. Because of this power stroke, what will happen, students? Imagine I come and hit your head. Your head will automatically move, no? What is on your head also will fall down. Same way, actin and myosin was bound. Let's say this is the actin head, this is the this is the myosin head, this is the actin. Myosin head was binding to the actin. Yes, ma'am. ADP and PI was there on the myosin head. Now they wanted to power stroke. They wanted to move. So when they moved, okay, when they moved out, the myosin's head skipped. Okay, this much space has come between the actin and myosin. So what happened, students? Here sliding happened. A slide. So actin just moved forward because breaking happened. No, divorce happened. Now the actin will move only. No, actin will move forward in life. Okay, so moving on, actin will move forward and myosin will go backside, because of which next ATP will come and bind. So here if you see, you can see sliding or rotation, that is where the power stroke happens, where the ADP and PI will be forced to leave outside, because of which myosin's head will move. Myosin's head will move. Clear? This myosin's head will move, leading to the breaking of the cross bridge. Okay, breaking of the cross bridge. Clear? That means the myosin's head moved and the cross bridge, that means marriage broke, students, divorce happened, that's all. Breaking of cross bridge happened. The moment cross bridge breaking happens, what will happen? What will happen? Okay, the next ATP will come and bind with the myosin. Do you want your muscles to always be like this only? You want your muscles to relax, you want your muscles to contract, all that has to happen tuck, 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 tuck. For the tuck, tuck, tuck to happen, sliding should happen, immediately ATP should come and join. Got it? Yes or no? So this is your sliding filament theory. Very simple. First hydrolysis of ATP will happen. After that cross bridge will happen. That means myosin and actin will bind. After that power stroke will happen. That means the ADP and PI will be forced to leave outside, because of which myosin head will move. That will cause sliding of actin. Actin will slide. Now think about your sarcomeres also. That is how the contraction will happen. Z line will move forward during this. Okay, during this. And after that, the immediate breaking, actin would have slided. Again, we need to release calcium for the troponin to get distracted. It's a long process. So by then the next ATP will come and attach, and the muscle contraction will go back to normal. Got it? This is how it happens. Okay. Now look here. Here you can see myosin head. Here ADP and PI will be formed. That will lead to what? Actin and myosin binding, cross bridge formation. Then students, you can see the ADP and PI will leave outside, because of which myosin and actin will separate. Okay, myosin and actin will separate, and here you can see as again the next ATP will come and join, again the cycle will keep continuing. Cycle will keep continuing. Yes or no? Clear? So now I'll show you that. I will show you that here. Okay, look here, the next one. Look here, students. See, it's a very fast process, very fast. See, ATP bound, okay, hydrolysis is, now they're separated, okay, separated. Now calcium got released, they got hydrolyzed, ADP and PI pushed out, because of which myosin head moved, and next ATP came and joined. Again, breaking is going to happen, ADP, PI, they will go and attach. Immediately they will do power stroke, again they'll move. So can you see actin is sliding and sliding and sliding and going. Okay, actin is sliding and going, myosin head also is doing this, 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 this. When I'm telling this means it's binding. Here it is going back. Okay, very fast process. How fast you see ATP is coming, joining, ADP, PI is getting released, sliding is happening. Okay, sliding is happening. Clear? Clear? Okay, chalo. That is your sliding filament theory. Okay, this is the most important thing, sliding filament theory. Done? Done? Students, you still have a chance to join the DNA batch. It is still running for 3,999. Okay, 3,999. After this, I will do one revision of this whole thing. And also, students, I have some surprise for you. And also, we have to do some PYQs. So, stay back. Students, DNA is still going to happen. That the surprise is still at 3,999. And if you want DNA plus NCERT long test series, you can go for 4,999. This is mainly for students who feel that, ma'am, even after I join a batch, I'm not a person who will solve properly, I need a proper test series, then this is the thing for you. And if you're a student who is like, no matter what, this 1 year I'm going to dedicate only for NEET, and I want the proper books, I want the year-long series, and I want a batch, then go for this. And if you're a student who is like, no ma'am, I just need a batch, I just need some guidance, and I need every day at least 5 hours of studying, pakka, then you can go for this. Okay, many students ask me, ma'am, which should I go for, which should I go for? Lot of confusion. So, this is one. Okay. Hi, Jay. This is the thing. Clear? This is the thing. Okay, and students, my DNA students are here, I hope. RNA students are already here. Students, we have a tug of war going to happen today. And I don't know whose team I'll be. Let's see. Depending on your people's vote, I'll be in one of the teams. Today we have a war between RNA versus DNA. For those students who have joined us newly, RNA is our free YouTube series and DNA is our paid batch, a very affordable batch for 3,999. And today it's going to happen on Vedantu NEET English. So at 4 o'clock, all of you quickly go and join the session. The link is already live. You can see RNA versus DNA. Diksha ma'am and Shreya sir is going to be one team. Me and Baswa sir is going to be one team. And we are going to have a tug of war with the portions that we taught you in both the batch as well as on YouTube. My portions are going to be human physiology portions, breathing and exchange, body fluids and circulation, excretion and locomotion, because these are the chapters I taught both the batches. So you're going to have questions from these chapters, and let's see who is going to win. Okay, so let's see whose batch I should be, whose group I should be. We are going to have some fun, students. It's going to be a very, very fun session. All my DNA students inform the other DNAs, and all the RNA students inform the RNA students. I am also going to put an official message on the Telegram as well. So today, do not miss it. And you know what? Should I reveal it or not reveal it? I'm thinking there is going to be a cash prize for the winner, and that cash prize is going to come in your Vedantu wallet. Vedantu wallet. And imagine you are a student who is planning to buy the batch, and with that cash prize, you're going to get so much less from this price. The cash price is going to be somewhere, can anyone guess? 500, 200, 300. Someone guess and let me know. Just guess. Let me see who is going to guess. Who is the one who is going to guess and tell me, ma'am, this is how much the cash price is going to be. Give it a guess, yeah. Jaldi. We have 2-3 minutes. Give it a guess. Come on. Oh, nice. RNA, ma'am. Let's see how DNA students are going to. If you people ask only, then only I'll come. Okay, otherwise I will not come. Cash price is going to be up to 1,000 rupees. That means, Amma, 20,000, 2,000. Students, it's going to be up to 1,000 rupees. Sometime it can go more also. And that money is going to come in your wallet. Very good, Hema. Perfectly right. It's going to come in your Vedantu wallet, and you can purchase anything for that price. Anything, whatever you like, you can purchase for that price. Okay, so do not miss it. The best student is going to get it. And students, please understand, every rupee that you earn, imagine you are earning it and you're going to tell your parents, Amma, you know what, I can get the batch for just 3,000 rupees because I earned the rest of the money. Okay, I earned the rest of the money. So do not ignore it. Every single money that you make by yourself is a reward, and especially when you use your knowledge and earn it, it's next level, students. And even if it's 10 rupees, you get a hats off from all the teachers and your parents, because you earned it with your knowledge, not because you did a physical labor, right? So it's very important. So definitely join today at 4 p.m. I will let you know the exact time on my Telegram, Biology by Gopika. So join the Telegram as well. And students, let's go back to our class. So today onwards, I am going to start a new thing which is called as GM Express. Okay, like your Chennai Express, Gopika ma'am's Express, where we will do a quick recap of whatever we studied in that class. Are you all ready? Are you all ready? I need some fire emojis in the chat. And if this is a success, if you people really like GM Express, every class we are going to have GM Express. Okay. Hasini, you will have a quiz. You will have a quiz. You will have to join the quiz and answer, and you will get it on the leaderboard. Okay, you will get it on the leaderboard. It's not like you answer on the YouTube. You will come on the Vedantu app. Okay, Vedantu app, and we'll do a V quiz. So there your leaderboard, fastest finger, all that will work. Okay, you will have questions of physics, chemistry, botany and zoology. Okay, students, ready for GM Express? So Gopika ma'am's express is going to start. That is your Chennai express. I'm going to start very fast. So students, what did we do today? We first understood the difference between locomotion and movement. Locomotion is when you do a displacement. Movement is when you do not do a displacement. It's not necessary to do the displacement. Okay. Now, the second thing that we understood was the properties of muscles, but before that we also understood the types of movement, that is amoeboid movement, ciliary movement, flagellated movement and muscular movement. Under amoeboid movement you have to remember two examples, WBCs and macrophages. Under your ciliary you have to remember all the linings, lining of the fallopian tube, lining of the respiratory tract, and under your muscular movements you can remember your jaw, your tongue and limb movement. All of these are muscular movements. You have around 639 muscles in your body. 40 to 50% of your body is covered by muscles and it is a mesodermal in origin. Okay, now next one is properties of muscle. Excitability, contractibility, extensibility and elasticity are the four properties of the muscles. The muscle has to get excited by a nerve impulse, the muscle has to retain its original position, the muscle has to contract, and the muscle should be having the ability to extend. Okay, extend. This is your four properties of the muscles. After we finish the properties of the muscles, we try to understand how does your skeletal muscle look like. So in your skeletal muscles, we understood there is a muscle. From the muscle, there is going to be your muscle bundles. Inside the muscle bundles, there is going to be muscle fibers. Inside the muscle fibers, there is going to be myofibrils. Now, your entire muscle is going to be covered by epimysium. Your entire muscle bundle is covered by your perimysium. Your muscle fiber is covered by your endomysium. Muscle fiber, the name comes fiber because it is long and slender. And then you have your myofilament or the myofibril, which has two contractile protein, actin and myosin. Actin is thin, myosin is thick. This causes the striation in the muscle. Now, based on the location, muscles are divided into three: smooth muscle, cardiac muscle and skeletal muscle, which you will be learning in animal tissues very easily. In this chapter, we will be only focusing on skeletal muscles. Okay, so skeletal muscles, you only have to remember fascicles, that is the muscle bundles, it's called as fascicles. These muscle bundles are joined together with the help of fascia, which is a connective tissue. Okay, now after that you understood epimysium, perimysium, endomysium being the innermost, which will be covering the muscle fiber. After that we understood about actin and myosin, which are the two contractile protein. Actin will lead to the thinner or the lighter bands and myosin will lead to the darker bands. One thick filament will be covered by 6 thin filaments and one thin filament will be covered by 6 thick filaments. All of you are following me? Am I too fast? Obviously express no, too fast. Okay, if you want me to write, it will take a lot of time, so I thought I will talk. Okay, then students, after that we went slowly into sarcomere. Now sarcomere is in the myofibril. It is the functional unit of the muscle that helps in contraction. Now what does sarcomere contain? It has two Z lines where the actins will be aligned. It has myosin where the M line will be holding it. And students, where only actin is present, it will be called as the I band. Where only myosin will be present, it will be called as the H zone. And where both myosin and actin will be present will be called as anisotropic band. Iso means same, aniso means different. So isotropic band and anisotropic band. And students, the H zone is the zone where only myosin is present. O zone is the zone where myosin and the actin is overlapping. You have Z line, M line, and Z line is the one that will push forward when a contraction happens. Okay, push forward when a contraction happens. Now, very clearly you have to understand few things is that your actin is the one that is going to contribute for your lighter bands. So that portion will be called as an isotropic band, and the portion where the dark bands are seen will be called as the anisotropic band, where both myosin and actin will be present. Got it? Now, one sarcomere has one A band plus one I band. Okay, half is there. We will calculate it together, one A band and one I band. Between both the Z lines is called a sarcomere. Both the Z lines together, that is from one Z line to another Z line, between is going to be a sarcomere. Okay, done. Okay, ma'am, understood. Now, I band, understood, A band, understood. Head zone is called as Henson zone. O zone is called as overlapping zone. Now, when a muscle contraction happens, which are the zones that are going to increase, which are the zones that are going to decrease? First of all, understand A band is not going to move an inch. A band is going to be constant. M line is going to be constant. I band is going to decrease in size. Head zone is going to decrease in size. O zone is going to increase in size because actin and myosin overlapping is going to be more. Overlapping is going to be more, and M line is going to be constant, A band is going to be constant. This shows that actin is the one that is sliding on the myosin, myosin is not sliding on the actin. Now students, the next thing is we have to study the structure of myosin, actin, and then how does the actin and myosin meeting happens, right? So Myosin, understand it is a monomer of meromyosin. It is formed with 6 polypeptide chain, 2 long chains and 4 short chains. It has light meromyosin chain and heavy meromyosin chain. Light meromyosin chain is formed because of the tails, and heavy meromyosin chain is because of the short arm and the head. Now on the head there is an ATP binding site and an actin binding site. ATP binding site is the site that will hide or mask the actin binding site. Now if you understand the myosin, you will understand it has a tail, it has a short arm, it has a head. Now this entire thing constitutes an entire myosin. Now from myosin, if you go to the actin, actin was first a globular actin, then it becomes a fibrous actin. In the fibrous actin there are 2 villains, troponin and tropomyosin. On the troponin there are 3 subunits: troponin inhibitor, troponin calcium and troponin by itself. Now troponin calcium is going to attract to the calcium from the neuromuscular junction to make the myosin binding site visible on the actin. Okay, that's how we distract the whole troponin and tropomyosin. Now students, when we move further, you will understand the actin structure, which is going to be like an infinity sign. It is actually a rope structure. In between you will have a myosin, tropomyosin, and you will have troponin. 3 subunits will be there. From there, you understand that the myosin binding site is actually masked by this troponin and tropomyosin. To distract them, we will be doing a neuromuscular junction, that is send a signal from the help of a motor neuron, which would be to contract the muscle. Now when the signal is sent, the acetylcholine will be accepted by the receptors on the neuromuscular junctions, and because of it an action potential will be created that will allow influx of sodium. Because the influx of sodium happens, automatically the calcium from the sarcoplasmic reticulum will be released. This calcium will go and bind with the troponin, that is TPC, and it will distract the troponin. Okay, it will distract the troponin and it will cause a conformation which will lead to the visibility of the myosin binding site. Now, since we made the myosin binding site visible, now our next job is to make the actin binding site visible on the myosin. On the myosin, the biggest problem is having an ATP binding site. So we will do hydrolysis of ATP, and hydrolysis of ATP will lead to generate ADP plus PI. Now when ADP and PI is generated, immediately cross bridge formation happens. That is where myosin and actin will bind with each other. That formation is called as the cross bridge or the marriage. Okay, actin and myosin cross bridge formation happen. Since ADP and PI cannot sit in myosin head for a very longer time, they will do a power stroke or remove themselves outside, because of which the myosin head is going to move and the actin is going to slide. The actin is going to slide. Because the actin slides, their sliding and rotation is going to be the third step. The first step was hydrolysis of ATP, which will form ADP plus PI. The second step was formation of cross bridge. The third step is sliding or rotation because of the movement of myosin head. And the fourth step is attachment of another ATP to the myosin head. Other ATP to the myosin head, because of which the muscle contraction has happened and relaxation has happened, and that is called as sliding filament theory. Sliding is called because actin slides over the myosin when the myosin moves, and that will be because of the power stroke and breaking of the cross bridge. This is the entire sliding filament theory. This will lead to the muscle contraction, and the next topic is going to be red fibers and white fibers. I hope GM Express was very clear. Gopika Ma'am Express is going to continue like this, where in 5 minutes I will be doing the entire revision of what we just did. I know it might be little fast and little breaking here and there, but let's try it. Okay, if it's helpful, then please let me know in the comments as well. Now students, are we ready for the question round? Okay, we are going to do a quiz time. Ready? Express over, quiz time. Vande Bharat, I thought Chennai Express you people did. Chennai, like that we'll make our own logo. I told it in one breath, now no breath. Okay, yes, quiz time, ready? Chalo, let's start, all of you. Students, definitely put the GM Express timing in the comments so many students get to know it, and students who just want a revision, they can watch that. Maybe they have to watch it in less more speed, but definitely let it in the comments, put the timing and tell GM Express starts from here. I want to hydrate myself, but there's no more water. I don't want to go outside now. So we'll finish this and then I'll go. Yeah, that's the song. Chennai, Chennai Express. Okay, that's what. Okay, students, look here. The heavy meromyosin is composed of what? Okay, the heavy meromyosin is composed of what? Globular head, short arm of meromyosin, globular head of meromyosin, short arm and tail, ATP binding site and tail. Oh, Naina was here. Naina did not see any message of yours. Yes, very good, very good. A is the answer, okay? A is the answer. Amazing. 4 points all of you got. Now, recognize the figure and find out the correct material. You have to find A, B and C. Look properly, okay? Look properly. This is your NCERT diagram, okay? So, look properly. A is there, B is there, C is there. Okay. Okay. Okay, I got the answer. I got the answer. I hope all of you also got. Yes, quickly. Don't make it too late. Yes, very good, very good. C is the answer. That is, this is going to be your C is going to be F-actin, B is going to be tropomyosin, A is going to be troponin. Okay. See, this is the use if you study with Gopika ma'am. Okay, I will show you the images also. That's why. So, which of the following about the globular head of myromyosin protein is not true? Not true. Okay, not true. It consists the active sites for actin. It has an active ATPase. This is what I told you, active ATPase. Otherwise, who will do the hydrolysis of ATP? It projects outward. It is a part of the light myromyosin. You have to tell which is wrong or which is not true. Okay. Yes, very good, very good. It is not a part of the light myromyosin, it's a part of the heavy myromyosin, right? It's a part of the heavy myromyosin. Those who haven't marked their attendance, students should mark it by now. Okay, done? Okay, I think, yes. In a contractile protein, each actin filament has, this is your homework question. Okay, this is your homework question. It's too long, no? So just write only the answer. Okay, just write the option. In a contractile unit, each actin filament has 2 F-actin polymer made up of F-actin monomer, 2 G-actin polymers made up of G-actin monomers, 2 F-actin polymers made up of G-actin monomers, 2 G-actin polymers made up of F-actin polymers. Full rap song it is, okay? Just put the answer in the comments, okay? So yes, students, done for the day, okay, done for the day. So like the video for the part 2, that is the skeletal system. The skeleton is asking you to like the video. So thank you so much for joining. I hope the session was really helpful. Let me know in the comments which was your favorite part of the class and also which was the favorite diagram. All this notes you will be getting on Vedantu NEET English Telegram. Do not fail to join us today at 4 PM for the biggest tug of war between our YouTube students and our DNA students. This will give you an idea how much dedicated we are on both the platforms and how much dedicatedly we teach on both the platforms. No discount, nothing at all, everything valuable, all valuable informations. So students, all you have to do is just like and comment, because that's the only way that is your Guru Dakshina for us, because we are not asking you any money. These videos are completely freely available on YouTube. I know multiple other teachers also are there, but you can understand the difference. You can watch the video. If it really helps you, then you should definitely put a comment and like the video as a Guru Dakshina for your teachers, because we every day read the comments and like the comments. So thank you so much for joining. Do not forget to like the video for part 2, so because skeletal system will feel bad. Next is skeletal system. Okay, thank you so much. Tata, bye bye, have fun. Homework in the comments.